Good morning everyone. Welcome back to the lecture series arranged by the Maratha Vidya Prasarak Samaj, SVKT College, Arts, Science and Commerce College, Deolali Camp, Nashik. I am your today's educator, Miss A.K.Eakhande, Department of Microbiology. As we all know that there are different types of the cells which are present in our body, and these cells contain different types of the organelles too. Along with the organelles, this cell contains different types of the proteins, and also it contains different types of the machineries which are play important role during the cell cycle or cell lifespan. Okay, this presentation explores the ribosome structure and types of the ribosome and functions of the ribosomes, as the ribosome is important and crucial role in the protein synthesis, emphasizing their significance in the cellular biology. As we know that there are different types of the cell organelles which are present in the cell, that is mitochondria, then ribosomes, and different types of the Golgi apparatus are also present in the cell. The endoplasmic reticulum has the two different types, that is the smooth endoplasmic reticulum and the rough endoplasmic reticulum. The smooth endoplasmic reticulum and the rough endoplasmic reticulum, out of that the rough endoplasmic reticulums have the structures called as an ribosomes. Okay, then the ribosome structure. The ribosomes are the smaller molecules. Also the ribosomes are dense and complex molecular machines which are found in all the living cells. They are composed of the two different types of the subunits. First one is the ribosomal RNA and the second one is the ribosomal proteins. The ribosomes are essential molecular machineries which is responsible for the protein synthesis in all the living cells. They are found in both the prokaryotics as well as into the eukaryotic cell. The ribosomes are present. Then the next one is composition of the ribosomes. There are, as we know that there are two types of the proteins which are present. The first one is the rRNA, that is the ribosomal RNA, and second one is the ribosomal protein. rRNA is the structural framework of the ribosome. The structure of the ribosome is given by the rRNA and act as an ribozyme, that is the catalytic RNA, to the catalyze the peptide bond formation during the protein synthesis. Second one is the ribosomal protein. This ribosomal protein help to stabilize the rRNA structure, and also it assists in the proper folding and functions of the ribosomes. Then structurally the ribosome consists of the two subunits. The prokaryotic ribosomes contains that is the 70S ribosome. In case of the prokaryotic, we having the 70S ribosome, the larger subunit and the smaller subunits which are present in the 70S ribosomes. The larger subunit is the 50S subunit. It contains the 23S and the 5S rRNA as well as the different types of the proteins which are present in the larger subunit, and the smaller subunit, that is the 30S subunit. The 30S subunit contains the 60S rRNA and the protein. Then the second one is the eukaryotic ribosomes. The eukaryotic ribosomes also contains the larger subunit as well as the smaller subunit. The larger subunit in case of the eukaryotic is the 60S, and in case of the prokaryotic, we are observing that the larger subunit is of the 50S, but in case of the eukaryotic ribosome, the larger subunit is of the 60S. It contains the 20S and the 5-S as well as 5S rRNA and the proteins, while in case of the smaller subunit, that is the 40S subunit, it contains the 18S and the rRNA and the different types of the proteins which are present in the eukaryotic ribosomes. The subunit composition: ribosome consists of the two subunits, that is the larger one and the smaller one. In case of the eukaryotic, the larger subunit is the 60S, while in case of the prokaryotic, the larger subunit is of the 50S. While the smaller subunit, in case of the eukaryotic, the smaller subunit is of the 40S, while in case of the prokaryotic, the smaller subunit is of the 30S. Each subunit is made up of the ribosomal RNA and the proteins, which together form the functional for the protein synthesis, and we can say that the translation process. The precise combination of the rRNA molecules and the ribosomal proteins, the characteristics and the function of each ribosome type. Ribosomes can be classified on the basis of their location and the function. First one is the free ribosome. These free ribosomes are found in the cytoplasm of the cell, which is important for synthesizing the protein for use within the cell. While in case of the second is the bound ribosome, these bound ribosomes are attached to the rough endoplasmic reticulum, and which is important for synthesizing the protein for secretion, and also which is important for the organelles like the lysosomes. Then the mitochondrial and the chloroplast ribosomes, which is found in the, if the ribosomes are found in the mitochondria, then we can say that the mitochondrial ribosomes, and if the mitochondria is present in the cytoplasm, then we can say that the mitochondria or the cytoplasmic or the chloroplastic ribosomes. The found inside the mitochondria and the chloroplast in the eukaryotic cell, the structurally resembles the prokaryotic 70S ribosomes, which is supporting the endosymbiotic theory, responsible for the synthesizing the protein required for the mitochondria as well as the chloroplast to be function. The ribosomal binding site, there are 3 different types of the sites which are present on the ribosomes. First one is the A site, that is the amino acyl site. In this amino acyl site, the binding of the upcoming amino acid tRNA carrying an amino acid. Then second one is the P site, that is the peptidyl site. This peptidyl site hold the growing peptide chain into the during the protein synthesis. Then third one is the E site or the exist site, which is important for the release of the empty tRNA after the amino acid has been added. These are the 3 main important sites, that is A site, P site and the E site, which is present in the ribosomal structure. This one is the structure of the ribosome. Okay, there are the larger subunit as well as the smaller subunit. Also it contains the E site, P site and the A site. The smaller subunit of the ribosome is attached, as well as the larger subunit of the ribosome is attached to the mRNA. Then the molecular structure. This site coordinates the tRNA binding and peptide bond formation during the protein synthesis. The ribosomes facilitate the translation process by accurate decoding message which is present on the mRNA sequence into the polypeptide, which is playing important role in during the gene expression. Then next topic is the ribosomal function or the ribosome function. The first and the main function of the ribosome is the protein synthesis. As we know that there is a central dogma when the DNA will convert into the RNA, that is the mRNA, and mRNA will convert into the proteins. The DNA molecule is converted into its own copy, that is the DNA will convert into the DNA with the help of the process called as a DNA replication, while the DNA molecule, that is the double stranded DNA molecule, will convert into the single stranded molecule, that is the RNA, which is the mRNA, and the process is called as an transcription, while the code which are present in the mRNA will translate into the proteins, and the process is called as an translation, that is the protein synthesis. The translation is also called as an protein synthesis. For the creation of the new generation, the genetic information which is stored into the DNA has to be transferred from one generation to the another generation. That is mean, if the DNA have one information, it should be transferred to the second generation with the help of the process called as an central dogma, where the DNA will convert into the mRNA and then the protein. The process beginning with the replication of the DNA followed by the transcription as well as the translation. Let us have the discussion on the translation process, that is the protein synthesis into the cell, and also the structure of the tRNA molecule, which is play an important role during the protein synthesis. The process by which the mRNA code for the particular protein is known as an translation. That is the code which are present on the mRNA, it should be converted into the particular protein with the help of the molecule called as an tRNA, because the tRNA having the anticodon while the mRNA having the codon which are present on the mRNA. In this process, the ribosome translates the mRNA produced from the DNA into the chain of the specific amino acids. This chain of the amino acid lead for the protein synthesis. Then it is process where the expenses of the ATP is required. That is this process, that is the translation process, required the ATP molecule, and this ATP is required and this energy is given by the charged tRNA molecule. The whole machinery of the translation is present in the ribosomes, that is the mRNA, tRNA, E site, P site and A site is also present into the ribosomal structure. Let's have a look on the structure of the tRNA. The DNA is transcribed into the mRNA on the basis of their complementarity. The translation is the process of conversion of nucleic acid information, that is the information of the DNA, into the amino acids. There is no complementarity between the amino acids and the mRNA. Hence, the translation is not controlled by the complementarity but by the genetic code which is present on the mRNA. Since the amino acid cannot read this codes which is present on the mRNA, we require the molecule called as an tRNA, because tRNA have the anticodons and the mRNA have the codons, that is complementary to the mRNA codons. This molecule, that is the adapter molecule which is responsible for the complementarity between the mRNA, the adapter molecule is called as an tRNA or the transfer RNA. Structurally, the tRNA is an inverted L-shaped molecule which has the anticodon loop and the amino acid acceptor end. This anticodon loop make the bases complementarity to the codons which are present on the mRNA and the amino acid end. Binds to the respective amino acid, thus it helps in the protein synthesis. Each amino acid has a specific tRNA and the initiation of the translation is with the help of the initiator codon, and the stop codon is responsible for the stopping of that process, that is the translation process, that we require the stops codon. This one is the structure of the tRNA. There are the 5' end and the 3' end. The 3' end contains the sequence called as an ACCC and also contains the discriminator bases, while it contains the T arm, D arm and the variable arm, and also the last one is the anticodon arm. This anticodon arm contains the complementarity between the codons which are present on the mRNA. Let's have a look on the translation process. There are 3 different steps. First one is the initiation, then elongation and the termination process. It begins with the small ribosomal subunit which is bind to the mRNA. The first step is what? The smaller subunit of the ribosome is binding to the mRNA molecule. Then the tRNA bring the specific amino acid to the ribosomes based on the codons which are present on the mRNA. The ribosome move along the mRNA, linking the amino acids in the correct sequence to form the polypeptide chain. Error during this process can lead to the dysfunctional proteins and emphasizing the precious of the ribosomal activity. This one is the diagram for the protein synthesis. First step is what? The transcription process, where the DNA molecule, that is the double stranded molecule of the DNA, will convert into the pre-mRNA. This pre-mRNA then get converted into the mature mRNA, and this mature mRNA will travel from the nucleus into the cytoplasm, that transported to the cytoplasm through the pore called as a nuclear pore. Then the smaller subunit as well as the larger subunit will bind to the mRNA, and the free ribosomes which are also binding to the tRNA. This tRNA brings the amino acid one by one depending upon the codons which are present on the mRNA and the anticodons which are present on the tRNA. Then after that, from the E site, the tRNA will leave after the complete protein synthesis will get happen. The growing chain of the amino acids are present on the tRNA itself. Then it leads to the formation of the new proteins. A role of the ribosome into the gene expression. The ribosomes play a important role in the gene expression by translating the genetic information coded in the mRNA into the functional protein. This process is essential for the cellular function, that is the cell to be function properly. We require the different types of the protein, and these different types of the proteins are functionally important for the gene expression. The growth and response of the environmental signal. Each protein synthesized serve a specific purpose within the cell, influencing the everything from the metabolic pathway to the structural integrity. The ribosome thus act as the bridge between the genetic information and the functional expression of the living system or the living organism. That is the genetic information which is coded in the DNA, and the functional expression, that is the protein which are synthesized by the translation process. Then the role of the ribosome into the cell growth and the metabolism. The ribosomes are essential for the producing the different types of the enzymes as well as the hormones and structural protein. Actively dividing cell, that is the cancer cells, have a higher ribosomal activity as compared to the normal cells. Then second one is the antibiotic targeting in the bacteria. Many antibiotics inhibit the bacterial ribosomes without affecting the eukaryotic ribosome. That is the antibiotics which inhibit the prokaryotic ribosomes, but they are not affecting the eukaryotic ribosome. Example is the tetracycline. The tetracycline is the antibiotic which bind to the 30S subunit of the prokaryotic ribosome and blocking the tRNA attachment. Then the second one is the chloramphenicol. This chloramphenicol antibiotic is inhibit the peptidyl transferase activity into the 50S subunit. Then second one is the advanced concept in the ribosome research. First one is the ribosomopathics, that is the disease related to the ribosome dysfunction. The mutations in the ribosomal component can lead to the different types of the disorder. Out of that, two are given here. First one is the Diamond-Blackfan anemia. This Diamond-Blackfan anemia defects in the ribosomal protein genes lead to the impaired red blood cell production. Then the second one is the Treacher's-Collins syndrome caused by the mutations affecting in the ribosomal biogenesis leading to the canifocal dysformalities. Then moving towards the conclusion of this session, the ribosomes are the fundamental to the cellular life, acting as the site for the protein synthesis and also play the important role for the translating the genetic information, understanding their structure and function, and essential for the comprehending the border between the biological process and the mechanism of the life. Let's have the figure out something. You have the 4 questions. The first one is the what are the ribosomes, then location of the ribosome into the cell. Third one is the 3 different types of the main steps, which are the main steps in the translation process, that is the protein synthesis. And you have to find that the what are the artificial chromosomes. Okay, thank you.